So in our last video, we covered membrane structure and components of the membrane that allow transport across the membrane. We looked at passive transport. Now we're going to look at active transport. So if you recall, passive transport moved with its concentration gradient and did not require energy. With active transport, we're going to see that we require a molecule of ATP. Remember, ATP is our cellular energy. And the reason that active transport requires this energy is because it's going to move substances against their concentration gradient. So a really important point of active transport is that it is moving things against the concentration gradient, requires a molecule of ATP, so it requires energy. And the purpose of this is to build the concentration gradient so then passive diffusion can actually work when it is needed. So here is a classic example, and the most frequent example you'll see of active transport, our sodium-potassium pump. So we take 3 molecules of sodium, we move them outside the cell using a molecule of ATP. Then we pick up 2 molecules of potassium, move them back into the cell, and then start everything over again. So with each round of our active transport, we move 3 molecules of sodium out, and we move 2 molecules of potassium in. And this is building the concentration gradient on either side of the membrane.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here's how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and move the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay, so let's review back over passive and active transport. Remember that passive transport moves with its concentration gradient, does not require energy. You can have simple diffusion where it can diffuse directly across the cell membrane, or you can have facilitated diffusion that uses either a channel protein or a carrier protein. Remember, channel proteins are an open tunnel through the cell membrane, whereas a carrier protein has to have a conformational change to take it. But all of these have their molecules moving with their concentration gradient, not requiring energy. Whereas our active transport requires a molecule of ATP, requires energy, and moves items against their concentration gradient. So our active transport, in a lot of cases, is responsible for building the gradient that will then be used by cells in a passive transport mechanism. So again, active builds the concentration gradient so that then passive transport can happen throughout the body. And this is important in our nerves and in our muscles. So animal cells tend to use that sodium-potassium pump that we just looked at, but another type of pump is our proton pump. And so plants, fungi, bacteria will use a proton pump. Notice also that hydrogen is a charged particle. Sodium and potassium were charged. Hydrogen is also charged. Remember, charged particles are ions. With hydrogen, if you recall, there is only 1 electron in the outer shell. If it has a plus charge, it means it has given away that electron. So it is now basically a proton, because that's all that's left, because it gave its electron away. So that's a proton pump. You're pumping hydrogens that have given their electron away and are pretty much just a proton. Again, the proton pump is using a molecule of energy. It is building a hydrogen gradient this time instead of a sodium and potassium gradient. But also notice the charged particles. So not only are we building a chemical gradient, but we are building an electrical gradient. So this is called an electrochemical gradient. We are not only building a concentration gradient of the molecule, but we are building an electrical gradient. So we are more positive outside the cell than inside the cell. And this is called our membrane potential. And membrane potentials are essential in our ability of our nervous system and our muscle cells to work. So by building the electrochemical gradient, when stimulus is received and then the charged particles flow back via passive transport, we have what we call an action potential. And then as the gradient is rebuilt, you will rebuild that resting membrane potential and be ready to make another action potential so that your nervous system or muscles can fire. Nervous system fire, muscles contract. And so this is our electrochemical gradient, built not only by our proton pumps, but also by our sodium-potassium pumps. Also due to gradients being built, and you can see this happen with sodium as well, is that when they flow back with the passive transport, flowing back with their concentration gradient, they co-transport and pull something against its concentration gradient. So it required the ATP and the building of that gradient to then flow back with its gradient, but it's pulling something in a build a concentration gradient, sucrose, across. So this is a co-transporter. This particular example is a symport. You can also have things move in opposite directions, which would be an antiport. So because it requires the gradient to be built by primary active transport, this is secondary active transport. It happens secondary to the primary active transport. Our last means of transport is vesicular transport. And we have endocytosis, where we bring things into the cell, entering the cell, endo, entering, E-N-E-N. Or we have exocytosis, things exiting the cell, E-X-E-X. So both are done via vesicular transport. If you recall an earlier diagram we were looking at, this showed exocytosis, release of molecules made in the cell, enzymes, hormones, things that the body requires to function, made in our endomembrane system, and then released via a vesicle out of the cell via exocytosis. They are secreted out of the cell. When we bring things into the cell via endocytosis, we can have three different possibilities. We have phagocytosis, phago, to eat, and that would bring in large food particles, bring in bacteria, things that need to be destroyed, be a phagocyte. And remember, phago, to eat, phagocytosis, to eat via a vesicle. Here you can see a amoeba eating a green algae cell, forming those pseudopods, pseudo meaning false feet, to form a vesicle around the algae cell and bring it in. With pinocytosis, pino means to drink. So pino, to drink in a solute dissolved in a liquid and bring it into the cell to be used within the cell. And here is a photomicrograph of pinocytosis, to drink. You also have receptor mediated, which is specific because you have to have a specific molecule binding to a receptor on the cell membrane. The binding of that molecule to those receptors, coating the plasma membrane there, will stimulate the formation of a vesicle to bring that molecule into the cell. So that is receptor-mediated endocytosis. So you have exocytosis, and then you have three mechanisms of endocytosis: phago, pino, and receptor-mediated.
Vesicles that form from the plasma membrane. Exocytosis is the movement of materials out of a cell by the fusion of vesicles with the plasma membrane. These processes enable patches of membrane to flow back and forth between the interior of the cell and the plasma membrane, reminding us that a cell is a dynamic structure. Exocytosis is the movement of vesicles in the cell to the plasma membrane. where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells that secrete enzymes into the digestive tract. In endocytosis, the plasma membrane invaginates or pinches in, forming a vesicle that moves the enclosed materials inside the cell. There are 3 types of endocytosis, each involving its own specific cell machinery. In phagocytosis, or cellular eating, the cell engulfs debris, bacteria, or other particles by extending pseudopodia around the material. Invagination produces a food vacuole. The material inside the vacuole will be digested after the food vacuole fuses with a lysosome containing hydrolytic enzymes. Phagocytosis occurs in amoebas and in immune cells such as macrophages and neutrophils in mammals. In pinocytosis, or cellular drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood. Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It's triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external receptor and its cargo are brought into the cell.
So an example of receptor-mediated endocytosis in humans is the ability of cells to remove low-density lipid proteins, which are our bad cholesterol, from the bloodstream, and this keeps you from developing atherosclerosis or buildup of cholesterol in the arteries. But there is a disease in humans called familial hypercholesterolemia, which lacks those receptors, and these individuals tend to have higher levels of LDL and develop heart disease earlier in life than most individuals.
Called good cholesterol and bad cholesterol. LDL, or low-density lipoprotein, is sometimes called bad cholesterol. It carries cholesterol that can stick to arteries, collect in the vessel lining forming plaque, and sometimes block blood flow. HDL, or high-density lipoprotein, is sometimes called good cholesterol. It takes cholesterol away from the blood and returns it to the liver.
So with exocytosis, all those molecules we make in the cell are sent out to signal adjacent cells. You can have local signaling where you go from cell to cell via the extracellular fluid. This is called our paracrine signaling. With the nervous system, we have release of a neurotransmitter via these vesicles, and they go into the postsynaptic membrane, targeting and moving that nerve signal from one axon to the next nerve cell. We can also have long distance signaling. This is usually with our endocrine cells, where a molecule, a hormone, is made here in the body, so in the brain, maybe the pituitary gland, released into the blood, travels via the blood to a distant target tissue, maybe in the adrenal cortex or in the testes or the ovaries, and starts a signaling series of events in those distance cells. So that's long distance cell signaling. When a receptor or a molecule binds to a receptor on a cell, we have 3 major steps in cell signaling. We have the reception, where it actually binds, the transduction of that signal from the binding through the cell, and then the response of the cell to that initial binding on the receptor. And so you can have responses such as activation of a gene or activation of an enzyme, although the enzyme would be made from a gene. Remember, a gene is a segment of DNA that codes for a protein, and as we've seen in our last couple chapters, how diverse proteins are in our bodies. So other types of receptors that we have that we activate, we have an ion channel receptor. This is where a signal molecule binds to a receptor and opens that channel and allows the molecules to flow in, and that is usually via facilitated diffusion, to where it flows with a concentration gradient and then causes a cellular response. Once that response reaches its target, then the ligand is released and the channel closes. Other types of signaling pathways we can have, this is a real simple signaling pathway, are things like a steroid hormone interacting with an intracellular receptor. And so a steroid tends to be a lipid molecule. And because it is a lipid molecule, it can diffuse directly across the plasma membrane, doesn't need to bind to an extracellular receptor as we saw in this last slide, but it can diffuse directly across and it'll bind to an intracellular receptor. And it sometimes can even bind directly to the gene. And so then the intracellular receptor is activated, comes and binds to DNA, activates a gene, and signals that transcription, the making of the messenger RNA, which is then transcribed into messenger RNA and then translated via our ribosomes into a protein. And so this is a signaling pathway using an intracellular receptor. Here are two more pathways that you can look at. This is cyclic AMP activation. So you have your first signal binding to an extracellular receptor such as epinephrine, stimulates the activation of a G protein, then activates adenylate cyclase, which is an enzyme, converts ATP into cyclic AMP, activates protein kinase, and this is usually an amplification. So this may be one molecule activated, this will be 5, this will activate 20, and so this is an amplification as it moves down this secondary messenger pathway. You can also have phosphorylation cascades, which are also amplified, that eventually result in the activation of a transcription factor. Remember, transcription is where you bind to a gene. A gene is a segment of DNA, codes for a messenger RNA. That messenger RNA is then coupled with ribosomes out in the cytoplasm, where it is translated into a protein.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
Okay, so this is our final talk on our transport across the cell membrane, looking at the active transport and signaling pathways. And as always, please let me know if you have any questions. Have a wonderful day.